Welcome to EPG Pathsala. Today I am going to discuss about the central dogma of life. In this, my talk will be divided into introduction to central dogma of life, history and the background information, what would be the flow of central dogma, DNA and RNA information, and finally summary. So the historical concept of gene is come from Mendelian inheritance, that is the law of inheritance, where the transfer of gene from one generation to other is based entirely on the phenotypic expression, where DNA is containing all the genetic information in the specific sequences of bases along the DNA double strand, which helps in the synthesis of proteins, which means DNA leads to specific traits by dictating the synthesis of protein, whereas protein are the connecting link between the genotypic and phenotypic expression. So again, to repeat, the central dogma, which is an important phenomenon that gives us an information regarding the flow of genetic information in the cells, where DNA is transcribed and produced messenger RNA that is ultimately translated and produced protein. This phenomenon also tells us how gene recognizes the sequence of mRNA molecules and thus specifies protein sequence. Since DNA is storage of all vital information and required to perform all key cellular function, and therefore cells have to preserve all the information intact in the DNA and copy it in the form of RNA. In short, DNA information is in the form of specific sequences of bases along the DNA strands. The DNA leads to specific traits by dictating the synthesis of proteins, where proteins are the link between genotype and phenotype, as I have already explained. Taken together, we can say that there are different processes or individual processes. For example, transcription and translation leads to the or transfer the genetic information for the conversion of one polymer to another, and there are different functions such as replication, that is the DNA is replicated from double-stranded, transcription, that is the formation of DNA to RNA, and translation, that is RNA to protein, which I will explain you in the later part of this talk. So let's talk about the dogma. What is the dogma? Meaning of dogma is that which one thing is true or something held as an established opinion. So initially, the dogmatic approach of DNA, RNA and protein was a bit different than what is in the new era of the molecular biology. So you can see here the prokaryote cell structure as well as the eukaryote cell structure, and DNA and RNA is coupled here in the prokaryotes as it lacks the nucleus. So the translation and transcription process is coupled in the prokaryotes, whereas in the eukaryotes, DNA is first transcribed into the mRNA, and that mRNA comes out from the nucleus to cytoplasm, that is a translocation from the nucleus to cytoplasm, and make the protein amino acid chain with the help of ribosome. So let's start with the history and background information. This is taken from the paper published by Francis Crick in the Nature volume 227 August 1970, where the central dogma enunciated by Crick in 1958 and the keystone of molecular biology ever since. So initially, works from the Dr. Howard Tamin say that DNA, RNA and proteins are self-replicating, and DNA produce RNA, RNA protein and protein to DNA, and vice versa. However, there were no experimental evidence have shown to establish this fact. Later on, it's a DNA, RNA and protein concept which come in action, where DNA is preparing DNA, DNA is transcribing to RNA, an RNA is translating to protein, and RNA is also self-replicating. However, this dotted line shows that it can happen in a special condition, like when the RNA is converted to DNA. This conversion requires reverse transcriptase enzyme and reverse transcription process. So this is the DNA, RNA and protein interaction, and this is the tentative and well-established fact in the molecular biology that how the flow of information from DNA to RNA and RNA to protein is achieved. Now you can see gene which is in the DNA sequence is transferred to the progeny in the formation of protein which has the functional role. So gene is now converting a protein for function, and this is the main source of inheritance or transmitting information from one generation to the progeny. If you decipher the central dogma of life, you can say that on a DNA stretch, transcription occurs and the mRNA produced. That mRNA translocates from nucleus to cytoplasm, and with the help of ribosome, it gives the stretch of polypeptide chain which fall down to the cytoplasm, and it gives information regarding the life. To elaborate the process of replication, which is double-stranded DNA to single-stranded DNA, is a process where double-stranded DNA to duplicate an identical copy, where this process perpetuates the genetic information. Next is transcription, where DNA is forming RNA, that is DNA to RNA, and in this process a segment of DNA that comprised a gene which read and transcribed into a single-stranded sequence of RNA, where RNA translocates from the nucleus to the cytoplasm for further function. Both DNA and RNA are come under nucleic acids that use base pairs of nucleotide as a complementary sequence, and with the help of enzymes it is producing DNA to RNA and vice versa. In the process of transcription, a DNA sequence is first read by RNA polymerase enzyme that produces an antiparallel complementary strand of RNA. For the study of gene expression, transcription is the first process where uracil of RNA is replaced by thymine of DNA wherever it is required. Moreover, a DNA molecule is transcribed into an RNA strand is called transcript, and sometimes uracil has been used as a regulatory RNA, whereas others encode one or many proteins. Furthermore, if a gene is transcribed which that encode a protein, messenger RNA is the resultant product of transcription, which is then further used during the generation of protein by a process called translation. Now come to the translation, which is RNA to protein, is a process through which mRNA sequences translated into amino acid sequence and joined together to form a polypeptide chain, otherwise known as protein. The synthesis of protein is initiated by mRNA with the help of large complex ribosome RNA. During the process of translation, ribosome recognizes a codon at a time from the RNA and then translates it into one amino acid with the help of transfer RNA. The function of tRNA in the translation process is to translate the codon sequence on the mRNA strand, where tRNA is to transfer a free amino acid from the cytoplasm to a ribosome and attach to the growing polypeptide chain until they reach a stop codon on the mRNA. So if you can go with the central dogma and omics, you can see genomics is the DNA which is transcribed to the RNA, which translated to protein, and with the help of different enzymatic reaction, it can give you the metabolites. So genomics, transcriptomics, proteomics, and metabolomics. And together with the transcriptomics and proteomics, it's called functional genomics. Elaborate the omics theory. The genesis behind omics theory is derived from the central dogma of biology. How to analyze the conserved and consensus sequence among the entire genome was a baffling issue in an ancient time. However, by the deciphering of the genetic flow, it was clearly indicated that genomics is a promising tool to analyze genome through gene flow, complexities in the genome, and of course population structure. We can also use genomics approach to understand many anomalies, for instance, single and double gene disorder, mutational analysis, conservative genomics, functional and comparative genomics across the multiple species. The peculiarity of genome is that base pair that code all genetic information of an individual, and after conversion of information from DNA to RNA, a process called transcription, transcriptome produced that carries all the information coding for a particular cell. For example, human kidney and heart have the similar genome but varied at transcriptome. And this is why kidney cells express genes that are required in the kidney function, whereas cardiac cells are expressing different genes to perform heart function properly. At the same time, transcriptomics are tissue-specific study based on the expression of selective or tissue-specific genes. However, transcriptomics can be regulated based on the physiological and pharmacological stimuli, and thus expression of genes are also modulated. Coming to the next point of the strength of omics is that the powerful approach of system biology, which is associated with other omics level, is proteomics. To understand the gene based on its protein expression, proteins are the building block unit of the cells and very important for the execution of function of a cell. In the end, it is also necessary to understand the end product of genetic flow, which is enabling us to deduce many interpretation and functionality of a gene, that is metabolomics. In this step, we can validate that what has happened during the genetic flow and also predict what could be the flow of information of a particular gene. Now come to the one gene, one enzyme hypothesis, because whatever we are doing, any function is governed by one gene. It was the initial hypothesis that one gene, one enzyme hypothesis, that in the formation of end product from the precursor, one enzyme is required, and that one enzyme is governed by a single gene. In case of any misinterpretation or any obstruction, the end product is disturbed. However, this one gene, one enzyme hypothesis was converted to one gene, one peptide and polyprotein hypothesis, that on the basis that not all the enzymes are protein, as some enzymes are also RNA in nature, for example, ribozymes. Further, this hypothesis is converted to one gene, one polypeptide hypothesis, where one protein may have more than one subunits. Exception to the rule is some genes do not produce different polypeptides, which is regulatory in nature, for example, in the 5' flanking region of a gene, and one gene may produce multiple polypeptide chains due to the splice variants. Now come to the nucleic acid sequences. Nucleic acid is the component of DNA which has the deoxyribose sugar base, which can be adenine, guanine, cytosine and thymine, and the phosphate groups. And the component of RNA is similar, but it has a ribose sugar instead of deoxyribose sugar base, where instead of thymine it has uracil, and RNA has also the phosphate group. So basically the nucleic acid had all the components except difference in the DNA and RNA is the ribose sugar as well as the thymine instead of uracil. The structural characteristics of DNA is double-stranded DNA, which means that it has two parallel and anti-parallel strands, and it connected through base pairing rules, which is the A is connected to adenine is connected to thymine, whereas guanine is connected to cytosine. And the structural characteristics of RNA, basically RNA is a single-stranded molecule. However, there are exceptions where there is double-stranded RNA is also available, and it's a limited base pairing. Now see the DNA as a building block. You can see that in the right-hand side, it's a nucleotides, which is linked to the phosphodiester bonds, and it's in anti-parallel direction. While in the right-hand panel, you can see purine and pyrimidine base, which is bound to the hydrogen bonding, and you can see A to T is the 2 hydrogen bond, where G to C is a 3 hydrogen bonding, and this is exactly in the anti-parallel direction. As I explained to you, the component of nucleic acid is nucleotide. And what is the nucleotides? Nucleotide is base, which is in yellow in color, sugar and phosphate, and you can see the color depiction in the all DATP, DCTP, DCTP and DTTP. So you can see with the color background that base, sugar and phosphates are making this deoxyribose components. The another important nucleic acid is RNA, and in RNA there are 3 types of RNA, which is the mRNA, and messenger RNA is complementary to information in DNA strand. It's different, variable in length, and it contains a specific structural information for the sequence of amino acid and is processed before using, which means there are total RNA. In the total RNA component, you will have mRNA, tRNA and rRNA, and before having messenger RNA, it has to be processed. Come to the transfer RNA, it's a multiple variety, each specific amino acid relatively smaller with a consistent 3D shape. A specificity for each amino acid is accomplished by triplet base pairing relationship between the codon on mRNA and anticodon of tRNA. I will explain you in detail what is the usage of the codon and the tRNA per se. Now come to the ribosomal RNA. Ribosomal RNA is structural component of the ribosome, the intricate machine that translates nucleotide sequence of mRNA into amino acid sequences of the polypeptide. Another RNA form is a small nuclear RNA. During the splice variant, you can have this, and this is structural component of the spliceosome, the nuclear structure that excises introns from the nuclear gene. I will explain you also in detail what is the architecture of gene, and over there I'll explain you how these introns are spliced off and give the mRNA. As I said in the beginning, that mRNA required a processing from the total RNA in existence. So basically DNA is transcribed to the messenger RNA. Messenger RNA carries the message to transfer RNA, and transfer RNA then translated to an amino acid chain which make up a protein. So the importance of nucleic acid is as follows. In the DNA or RNA, there are 4 different bases are present in the monomeric form and act like later to the alphabet, which communicate the information from one generation to another or in the expression per se. The specific sequence of hundreds or thousands of nucleotides in each gene carries the information for the primary structure of a protein, and the primary structure of protein is the linear order of the 20 possible or 21 possible amino acids. And in order to obtain the information from DNA depicted in one chemical language to protein, protein is deciphered into the another and require 2 major actions, which is transcription and translation. Basically, translation is a process which is in the 5' to 3' direction, and one strand acts as a template, and the template strand is basically run from 3' to 5' direction, whereas the transcription of a gene product produces a messenger RNA molecule, and mRNA carries the message from nucleus to cytoplasm for the ribosome. During the translation process, the information contained in order to nucleotides in mRNA, which is used to determine the amino acid sequence or polypeptide. So translation is occur mainly in the cytoplasm with the help of ribosome. So the bridge between the gene to protein is transcription and translation. Why I have said this? Because gene dictate to make a specific protein, as we have already seen in the one gene one enzyme hypothesis, as well as one gene one protein and one gene one polypeptide hypothesis, that every gene is responsible for the production of a particular protein. The bridge between DNA and protein synthesis is RNA, which is the messenger RNA, and RNA is chemically similar to DNA except uracil base, because the thymine is absent in the RNA. RNA molecule usually single-stranded in a structure. However, there are exceptions where you can find double-stranded RNA as well. To summarize my talk, its concept of gene derived by the Mendelian inheritance. DNA is double helix with complementarity base A to T and C to G, and the biochemical process that read up the DNA from the 5' direction towards the 3' direction, which is the replication and transcription. A gene can be located either on the plus strand or on the minus strand, which is also called the sense or antisense strand. DNA information is copied over mRNA that acts as a template to produce proteins. So take home message for Central Dogma chapter is what we learned: a genetic flow of gene, that is from DNA to RNA and RNA to protein and protein to the metabolic products. Moreover, we also learned today that what is the approach of ancient versus modern central dogma approaches and hypotheses. Next, we learn how the replication, transcription and translation processes are very intricate and utilized by a gene to make a genetic flow. Then we have learned omics theory, and in this omic theory we came to know the existence of genomics, transcriptomics, proteomics and metabolomics. Then we further understood that how one gene, one enzyme hypothesis came in existence, followed by the different nucleic acid components and the structural architecture. We have also learned that how DNA is varied from RNA and different types of RNA such as messenger RNA, ribosomal RNA and tRNA and a small nuclear rRNA. And finally, we conclude this chapter that the first step of genetic flow is transcription, which occurs in the nucleus, where one strand acts as a template for the synthesis of complementary RNA strand, whereas the end product is mRNA that carries the message from nucleus to ribosome. Moreover, we have also learned that how the reverse transcription process comes in existence, where DNA to RNA, instead of DNA to RNA, RNA to DNA we reached, and on contrary to the translation, which is the last step of the genetic flow, occurs in the cytoplasm with the help of ribosome, and the end product of translation is protein. Further, we have also analyzed the concept of gene is classically defined based on the Mendelian inheritance, that how it transmits from one generation to another, like from progeny to offsprings. To summarize, gene program protein synthesis via genetic messenger, and that is mRNA. The molecular chain of the command in a cell is DNA to RNA and RNA to protein, and this is referred as the central dogma of life. Thanks for your attention.